Hi, guys, and welcome to chapter 22 on the digestive system. There's kind of three lengthy parts to the digestive system. I've included all of them because a lot of it is anatomy review, and I'll try to point out the specific physiology stuff that I want you guys to work at too. The main functions of the digestive system are seen here. Basically, it's taking in food and breaking it down to nutrient molecules that can be absorbed into the bloodstream and then eliminating any wastes that the body's digestive system cannot digest or ingest. The organs of the digestive system fall into either the alimentary canal, also known as the GI tract, the gut. This is the continuous muscular tube that runs from the mouth to the anus. This canal will digest the food, absorb fragments through the lining into the blood, and the organs are listed there that make up this one continuous long tube. And then the other part of the digestive system organs are the accessory organs. These are not a part of the tube, but they will secrete substances into the tube or help with digesting the food particles in a mechanical way. So your teeth, your tongue, your gallbladder secretes bile. The digestive glands will produce secretions that break down food, like your salivary glands break down starch and carbohydrates, and the liver and pancreas will secrete other digestive enzymes to help break down food. So here's a good kind of review of the structures that make up the alimentary canal and related accessory digestive organs. And you can see these here. Take a moment to just pause this slide and make sure you can label everything on this picture. The processing of food involves 6 essential activities: ingestion, propulsion, which is the movement of food. That consists of swallowing, peristalsis, which involves alternating waves of contraction of smooth muscle, and relaxation to kind of squeeze the food along, kind of as if you're squeezing a Go-Gurt tube of yogurt. And here's a look at the differences between peristalsis and segmentation. So peristalsis again, uses smooth muscle to kind of contract and relax and push the food down. This primarily is propulsive. Some mixing could occur. So food is always moved distally along the tract. In segmentation, you have non-adjacent segments of the alimentary canal organs contracting and relaxing. So you have food moving forward and then backward. So we have this mixing of food, breaking it down, a little bit of pushing it forward, but kind of mixing, stopping, pushing forward, pushing back, and that's segmentation. The mechanical breakdown of food involves chewing, mixing food with saliva, churning food in the stomach, and segmentation, which we just talked about. Digestion are all the catabolic steps that involve enzymes breaking down complex food molecules into their chemical building blocks. So we're taking food and breaking it down into its simplest components to be used by your body cells or to be absorbed into the bloodstream. Absorption is the passage of digested fragments from the lumen of the GI tract into your blood or lymph, and defecation is the elimination of indigestible substances via the anus in the form of feces. So here's just a look at the GI tract activities and where each of these activities occurs along the GI tract. And I'll let you guys review that on your own too. So the organization of the digestive system involves the peritoneum, the peritoneal cavity. This is just the relationship of where your organs fall within the peritoneum. The peritoneum is the serous membrane of the abdominal cavity that consists of a visceral and parietal peritoneum. And these are just membrane layers that will either cover the surface of the digestive organs or line the body wall. The peritoneal cavity is the fluid-filled space between the two peritoneums, and the fluid will lubricate the organs that are able to be moved. The mesentery is a double layer of peritoneum. The layers are fused back to back. It extends from the body wall to digestive organs. Your mesentery provides like a route or an area for blood vessels, lymphatics, and nerves to travel through. It holds all the organs in place and also stores fat. And your intraperitoneal organs are organs that are located within the peritoneum. Retroperitoneal organs are located outside or posterior to this peritoneum membrane. These are your pancreas, your duodenum, and parts of your large intestine. I won't ask you guys to know specifics about what organs are what. I'm just kind of giving you an overview of what these words peritoneum, mesentery mean And you can see kind of where these organs are located and what I mean when I talk about the parietal and visceral peritoneum, and which organs are located out of it, retroperitoneal, or inside of it, intraperitoneal. Peritonitis is just inflammation of this peritoneum membrane. It could be caused by a piercing wound, an ulcer, ruptured appendix. It's dangerous and lethal if it becomes widespread, and the treatment for it is to remove the debris and give a lot of antibiotics. The histology, so all of the digestive organs have the same four basic layers or tunics, we call them: the mucosa, submucosa, muscularis, externa, and serosa. And I do want you guys to know these four layers and the order that they go in. So the mucosa, it lines the lumen. So this is the innermost layer. It functions in secreting, absorbing, and protecting kind of the inner layer of the lumen of the digestive canal. And it's made up of these three layers, which isn't super important, but I want you to know that the mucosa is the innermost layer. So it's what all the foodstuffs will touch as they travel down the GI canal. This is the epithelium that makes up. It's a mucus secreting simple columnar epithelium. It secretes mucus to protect the digestive organs from enzymes and also ease food passage. It also could secrete enzymes and hormones if it's in the stomach and small intestine. The lamina propria is just a loose areolar connective tissue, and a muscularis mucosa is a thin layer that produces local movements of the mucosa within this first mucosa layer. So here is the look at the mucosa layer. And again, this is the innermost layer that will line the lumen tube. And these are the three kind of parts of the mucosa that we talked about. Underneath the mucosa, and we'll talk about these, but I'm going to point out this picture. I think this picture is actually in the quiz. So you do want to understand how to label a picture like this. The submucosa is the layer just kind of more superficial to that mucosa layer. The muscularis externa layer has two kind of areas of muscle, the circular layer and longitudinal layers. So these are just layers of different smooth muscle. And then the serosa layer is the outermost layer of the GI tract. It's made up of connective tissue and epithelium that helps anchor kind of the digestive canal to wherever it is in the body. So here's the submucosa layer. It consists of areolar connective tissue. It'll contain all the blood and lymphatic vessels that supply the surrounding GI tract tissues. The muscularis externa layer, this is the layer that's responsible for segmentation and peristalsis, so moving or mixing the food along. And it contains these two kind of smooth muscle layers. The circular layer of the muscularis externa will thicken in some areas to form sphincters. And if you remember from anatomy, these sphincters will open and close to allow the food particles to pass through as they're going on to the next spot in the GI tract. The serosa is then the outermost layer, and that's really all you need to know. That it's kind of a little different as it travels throughout the GI tract, but this is the serosa. So again, this is the basic structure in these four tunics or layers of the alimentary canal. Blood supply is in what we call splenic circulation, which includes arteries that branch off the aorta to serve the digestive organs. You can see those there. The hepatic portal circulation will drain nutrient-rich blood from the digestive organs and deliver to the blood for processing. So when you eat food, it gets broken down into its chemical building blocks, and those chemical building blocks or molecules get absorbed into the bloodstream. The bloodstream takes it through the hepatic portal circulation, where your liver will process and detoxify all of those chemical breakdowns of the food that are being digested. So your liver is kind of the major cleanup part of the body of everything you eat. Control of the digestive system. I'm actually going to skip this part. It has to do with the nervous system. It's referred to as the enteric nervous system. So the GI tract has its own specific nervous system, also known as the gut brain. And it contains more neurons than the spinal cord. Outside of that, I won't ask you anything else about the enteric nervous system. It's just the GI tract nervous system. Still controlled by the brain, but we're going to skip over a little bit of that. Functional anatomy of the digestive system. These will start, and this is kind of where the rest of the slides go through, including parts B and C. I'm going to move quickly over the anatomy of it. So here's our mouth and associated organs. You should know that mouth is where food is chewed and mixed with saliva that contains salivary amylase, an enzyme that helps to start the process of digestion. Here are the associated organs of the mouth. You can look through the oral cavity, what makes up the oral cavity, what lines it. The mouth is lined with stratified squamous epithelium to help resist abrasion of all the food. Lips and cheeks. I'm not going to ask you guys too much more about, details about this, but you should know just the general anatomy of the oral cavity or the mouth. The palate forms the roof of your mouth. The hard palate is the more anterior side. So the part that you could probably touch with your tongue, that's the hard palate. It's made up of the palatine bones. And then the soft palate is formed mostly of skeletal muscle that's in the back. It will raise and close off the nasopharynx during swallowing so that no food and drink goes up through your nose. Then the uvula is the finger-like projection, this kind of boxing bag that hangs down at the end. And the uvula and soft palate will raise up when you swallow to kind of close off the nasopharynx opening. The tongue occupies the floor of the mouth. It will help to kind of grip, reposition, mix the food during chewing. It'll form the bolus, which is the mixture of food and saliva. It also helps to initiate swallowing, speech, taste. The lingual frenulum is the attachment to the floor of the mouth. Here are the papillae, which are peg-like projections of underlying mucosa. I covered this in anatomy, so I'm going to skip over papillae. You guys don't need to know that. We'll skip over kind of some of this, but papillae are just the bumps where that house all your taste buds, and that's your tongue. Tongue-tied, fused tongue. This is surgical snipping. This has sometimes happened with children who are born with an extremely short lingual frenulum. We'll skip over that too. So saliva, most saliva produced by your salivary glands is produced outside of the oral cavity. You still have some minor salivary glands. The big thing I want you to know about saliva is that it begins breakdown of starch with the enzyme amylase. Here are the major salivary glands. The parotid lies on your masseter. The submandibular is below the mandible, and the sublingual salivary gland is underneath your tongue. So here's a look at the salivary glands, the three major ones, and where they are located. And they all have ducts that bring their enzymes to the mouth. Here's just a look at the different cells that make up the salivary glands. You can see that they're secreting cells because they're secreting substances. These serous and mucus cells, some will secrete enzymes. The mucus cells will produce mucus. Xerostomia is dry mouth. Lack of moisture may lead to difficulty with chewing and swallowing. This can be caused by different medications, diabetes, HIV, and Sjogren's syndrome, which is an autoimmune disease affecting other moisture-producing glands throughout the body. So here we have saliva. It's mostly water, slightly acidic. It's made up of these electrolytes. Again, the big thing I want you to know is it's made up of this salivary amylase and lingual amylase. There are some lysosomes and defensins, which just help to protect against any microorganisms. And here's a video you can watch of the salivary glands to show you where they're located. The control of salivation, about 1,500 milliliters a day, can be produced. We're not going to go too much into the control. It is under parasympathetic response. Remember, the parasympathetic system is the rest and digest. So your parasympathetic system will initiate salivation. Decaying primary teeth can be painful and could lead to an infection. We'll kind of skip over this as well. Teeth, I won't ask you too much about. They help with mastication, which is the process of chewing that grinds the food into smaller fragments. Dentition and dental formula, this takes you through when teeth are formed and when they come in. We'll go a little bit through kind of the anatomy of the teeth. So the crown is the exposed part above the gum, and it's covered by enamel, the hardest substance in the body. The root is the portion embedded in the jawbone and it's connected to the crown by the neck. And you have different types of teeth, the canines, incisors, and premolars, and then the molars. That's pretty much, I think, all I'm going to get to. Let's just take a look at a picture of a tooth. This is just the general anatomy of a tooth you can review. Here's the root with the pulp cavity containing all the blood vessels and nerves within it. You can see the gingiva are the gums, And then the neck connects the root to the crown, which is the part that is exposed, the part that you brush. And it's covered by the enamel, which is the hardest substance in the body. And dentin is just the layer below the enamel. Okay, this is kind of interesting about an impacted tooth that you can read about, but I won't ask you guys questions on that. Cavities is any sort of demineralization of enamel and dentin from some sort of bacterial action. A dental plaque is usually sugar, bacteria, or debris that adheres to the teeth, and acid can dissolve the calcium salts, which could digest the organic matter to try to demineralize or basically wear away the tooth itself. And obviously, floss and brush twice a day for that. Gingivitis, again, it's an itis, so we have some sort of inflammation of the gums. It's usually due to plaque calcifying to form a tartar. This will disrupt the seal. Anaerobic bacteria can infect the gums, and the infection is reversible if this tartar is removed. Periodontal disease, again, if you're studying, any of you are studying to be an assistant or want to go on to be a dentist or a cleaning assistant, I'll let you guys kind of read through that on your own. So I'll stop this here and we're going to move on to the second part, and give me a minute to pull it up, or feel free to fast forward the PowerPoint as I pull this up here. Okay, so we're in part B now, and let me go ahead and make sure my screen is shared. So again, another kind of lengthy part. In part B, we're just going to kind of keep moving down. The pharynx, esophagus. The pharynx is divided into your oropharynx and laryngopharynx, which share the passage of food, fluids, and air. You have different muscles that help to the food to become swallowed down as it gets to the esophagus. The esophagus itself is a flat muscular tube. It pierces the diaphragm at what we call the esophageal hiatus, and it joins the stomach at the cardial orifice. The gastroesophageal sphincter surrounds the cardial orifice. It helps to keep it closed when food is not being swallowed, and mucus cells on both sides of the sphincter help protect the esophagus from acid reflux because the stomach is very acidic. Esophageal glands will secrete mucus to aid in the movement of bolus, and you have different muscular walls to help move the bolus of food down as well. So you can see here the 4 different tunics or layers of the GI tract of the stomach, and how they kind of squeeze the food down so it'll be a flattened tube. Heartburn is caused by stomach acid regurgitating into the esophagus. It's the first symptom of gastroesophageal reflux disease, and it's usually caused by excess food and drink, extreme obesity, pregnancy, running. It also can be caused by a hiatal hernia, which is where the stomach protrudes, or a part of it, above the diaphragm. That is bad because it can lead to an inflammation of the esophagus, an ulcer, or even cancer of the esophagus. So here we go in the deglutition is the process of swallowing. You have 22 different muscle groups that help you swallow food down. You don't need to know all these phases, just know that there's a lot of muscles involved in swallowing the food. It takes about 6 to 8 seconds for the bolus to travel down your esophagus into your stomach. So here shows deglutition, swallowing into the stomach. Peristalsis will move the food through the esophagus and into the stomach. The gastroesophageal sphincter will open then to allow the bolus to enter the stomach. And you can see here how the muscles get relaxed towards the top, so the bolus continues down. So this leads us to a stomach. It's a temporary storage tank that will start the chemical breakdown of protein. It converts the bolus of food to a paste-like chyme. It has a volume of about 50 milliliters, but it can expand to 4 liters. And when it's empty, the stomach mucosa forms many folds called rugae. Here are the parts of the stomach that I'm just going to kind of pass over. So pause the slide here and review these anatomy parts of the stomach and make sure you're familiar with all of that. And here's a look at the anatomy of the stomach, kind of a cadaver view to show you where the liver is, the lesser curvature, greater curvature, and how the spleen kind of sits out to the right. Okay, so I'm going to pass over the kind of some of the anatomy. The greater omentum drapes inferiorly from the greater curvature over to the intestine. It blends with what we call the mesocolon, and it contains fat deposits and lymph nodes. We sometimes call this the apron. It anchors the large intestine to the abdominal wall, so it just kind of keeps everything in place. The autonomic nervous supply supplies the stomach with sympathetic and parasympathetic fibers, and it has its own blood supply from the celiac trunk and veins in the hepatic portal system. It has the four normal tunics that we talked about, but the muscularis and the mucosa are modified because the stomach is doing different churning and mixing actions than the esophagus. The mucosa layer has what we call gastric pits, which lead into gastric glands. And these gastric glands will produce gastric juice that help with the digestion. The mucosa layer also contains more alkaline rich mucus that helps to kind of counteract the acidity of the stomach. So here's a look at the mucosa layer and how it has these gastric pits in them and the other layers of the tunic found below. Here are different types of gland cells. These are glands in the fundus and the body of the stomach that produce most of the gastric juice. The glands include secretory cells, and I do want you to know these secretory cells because they're really important for some of the digestion. The mucous neck cells, parietal cells, chief cells, and enteroendocrine cells. And all these different cells, you can see that they line these gastric pits, and they all secrete a different substance to help to absorb or digest foodstuffs that get trapped in these gastric pits. The mucous neck cells secrete thin acidic mucus, which we don't really know why. The parietal cells, the big one these guys secrete are the hydrochloric acid, which denatures protein, activates pepsin, and breaks down plant cell walls and kills a lot of bacteria. So your stomach is a very acidic environment due to your parietal cells secreting hydrochloric acid. Intrinsic factor is also secreted by your parietal cells. And this is just a glycoprotein that's required for the absorption of vitamin B12 in the small intestine. So intrinsic factor is just very important for your small intestine to be able to absorb a vitamin B12. Chief cells secrete pepsinogen, which is an inactive enzyme that will be activated to pepsin by hydrochloric acid and by pepsin itself. It's a positive feedback mechanism, and chief cells also secrete lipases, which will digest about 15% of lipids. Enteroendocrine cells secrete chemical messengers. They act as paracrines, so they secrete serotonin and histamine, and they also secrete hormones like somatostatin and also gastrin. So just understanding that your enteroendocrine cells just secrete different paracrines, which are different molecules, and also hormones. And that's pretty much all you need to know. I mean, this is probably a great picture just to review. The parietal cells secrete HCl and intrinsic, chief cells secrete pepsinogen, and the enteroendocrine cells secrete hormones and paracrines. And that's pretty much what I want you to know from that. The mucosal barrier protects the stomach, and it's created by three different factors. And there's a harsh digestive conditions within the stomach because it's very acidic with that hydrochloric acid. So the mucosal barrier just keeps this lining of the stomach protected from acid. Anything damaged will be quickly replaced by different epithelial cells every 3 to 6 days. Gastritis is an inflammation caused by anything that breaches the stomach's mucosal barrier. You can have a peptic or a gastric ulcer, which could cause an erosion in the stomach wall. Most ulcers are created by a bacteria, but also can be created by NSAIDs such as aspirin. So that's one thing you have to remember, not to take too much aspirin because it's kind of wearing away this mucosa layer wall. That's a look at a gastric ulcer. If any of you had an ulcer, I think it's incredibly painful. Here's the bacteria that most commonly causes gastric ulcers. Again, these bacteria just kind of eat away at the mucosa layer of the stomach to try to get through it. The process is carried out by the stomach. This is just a good kind of summary. It carries out the breakdown of food, serves as a holding tank for food, delivers chyme to the small intestine. It denatures proteins by that hydrochloric acid, so it makes proteins unable to kind of work anymore. It's just trying to break them down. And then pepsin carries out enzymatic digestion of proteins. And that's what pepsin does. The lipid-soluble alcohol and aspirin will be directly absorbed into the blood at this point. So that's why you feel the effects of alcohol and aspirin, for that matter, very quickly because there are two substances that get absorbed into the blood from the stomach. Most, if not the rest of everything else, gets absorbed when it gets to the small intestine. The only stomach function essential to life is the secretion of intrinsic factor for vitamin B12 absorption. Vitamin B12 is needed for red blood cells to mature, and lack of intrinsic factor causes pernicious anemia, which is an autoimmune disorder, and you can be treated with B12 injections. The gastric mucosa secretes more than 3 liters of gastric juice per day. It's regulated by neural and hormonal regulations. Gastric secretions are broken down into 3 phases: the cephalic, gastric, and intestinal phase. I won't have you guys know these. These are just different phases when gastric secretion will be secreted. Okay. And we'll kind of look at this kind of as a final summary of the neural and hormonal mechanisms that regulate the release of gastric juice. Again, I won't have you guys know all these phases, but there's different stimulatory events, whether the sight and thought of food, the stomach distension activates stretch receptors, and also inhibitory events that will inhibit this. Remember, your parasympathetic is your rest and digest division. So decreased parasympathetic activity will inhibit stomach activity or inhibit gastric juice secretions. Seeing or thinking or smelling food will stimulate gastric acid juice secretions. Here are different hormones and paracrines that act in digestion. Again, maybe being familiar with the names is a good idea, but an understanding exactly what they all do, I won't have you guys know. Okay, parietal cells pump hydrogen. This is the mechanism of how parietal cells create hydrochloric acid. I'm not going to ask you guys to know the mechanism. I just want you to remember that parietal cells are the ones that create and secrete hydrochloric acid. And this takes you through that mechanism. Again, parietal cells, they contain many mitochondria because there's a lot that goes into secreting this hydrochloric acid. And that's all I want you to know for that. Response to the stomach to filling, you have a receptive relaxation, gastric accommodation, and then gastric contractile activity. Again, this is kind of the regulation of how quickly things move through the stomach and then how the stomach is emptied. Contractions will be most vigorous and powerful near the pylorus, which is the bottom inferior end of the stomach. 30 milliliters of chyme produced is either delivered in 3 milliliter spurts to the duodenum, so about 3 milliliters at a time is passed on to the small intestine. The rest is forced backward into the stomach to continue the process of storing it or absorbing, digesting, breaking it down. So this is a look at propulsion waves that move from the fundus toward the pylorus. And I'm going to go ahead and just keep going with that. Sorry, I just got a notice up on my screen. Grinding is the most vigorous peristalsis moving. These are just different peristaltic waves that take food 3 milliliters at the time, gets through the pyloric valve. The rest is what we call retropropulsion, where the peristaltic wave closes it, forces most of the contents of the pylorus backward into the stomach. So those are just the waves in the stomach that keep the food mixing and eventually emptying. So eventually the stomach will empty into the duodenum, which is the first part of the small intestine. The stomach will empty in about 4 hours, but it'll increase in fatty chyme entering the duodenum that they increase the time to 6 hours or more. Okay, and then neural and hormonal factors that inhibit gastric emptying. Vomiting emesis is caused by extreme stretching, also by some sort of irritant like a bacteria, a toxin, excessive alcohol, spicy food, or certain drugs. Excessive vomiting, the big thing you want to worry about could lead to dehydration, but also electrolyte and acid-base imbalances leading to alkalosis. Then we'll get to the liver, gallbladder, and pancreas. These are all accessory organs, so they are not part of the GI tract. The liver is the digestive function of that is to produce bile, and the bile is what we call a fat emulsifier. So it surrounds fat and breaks it down in a process known as emulsification. The gallbladder just stores the bile. So people can live without their gallbladder because the liver will continue to produce bile. The gallbladder just stores bile. And then the pancreas supplies most of the enzymes needed to digest chyme, as well as bicarbonate to neutralize stomach acid. The gross anatomy of the liver, it's about 3 pounds. There's different lobes to it with different ligaments, which those are seen here. You can review the anatomy of the liver a little bit on your own. And then the gallbladder kind of sticks out or hides out on the inferior side of the liver. The inferior side of the liver is seen here with a gallbladder right located there. The hepatic artery and vein will enter the liver at what we call the porta hepatis. The bile ducts, the common hepatic duct leaves the liver. The cystic duct connects to the gallbladder, and then the bile duct is formed by the union of the common hepatic and cystic ducts. Those ducts, I do want you to know, I kind of stress that importance in anatomy too. The gallbladder is connected to the cystic duct, and then the common hepatic duct is what will travel into the different lobes of the liver. And then the bile duct, also known as the common bile duct, is the connection of the common hepatic duct with the cystic duct. The liver lobules, they're composed of plates of hepatocytes, which are liver cells. They're in a hexagonal structure. There's a central vein located in the longitudinal axis of each kind of plate of hepatocytes. There's a portal triad that includes a hepatic artery, a hepatic portal vein, and a bile duct, which receives bile from the canaliculi. So the hepatic artery will supply oxygen to the liver, and the hepatic portal vein brings the nutrient-rich blood from the intestine. Remember, the liver gets all the blood that came directly from the GI tract. So anything that gets absorbed from the GI tract will go through the liver, and it enters the liver via this hepatic portal vein. The hepatic artery also enters it to supply oxygen that it needs, and the bile duct will receive bile from the bile canaliculi, which will be what produces bile. So here's this hexagonal lobule, which is made up of all the cells of the liver, the hepatocytes. So it's kind of cool. They're arranged in this hexagonal shape. Here's the central vein that's bringing the nutrient-rich blood from the digestive tract. And here's just connective tissue surrounding the lobule. Liver sinusoids are leaky capillaries located between the hepatic plates. And then the liver has its own macrophages, which remove debris and old red blood cells. And this is just kind of look about how this hexagonal shape is arranged with the central vein and how it's bringing nutrient-rich blood to the liver, to all the hepatocytes, which are detoxifying it. So the plates of hepatocytes, you have these bile ducts receiving blood from bile canaliculi. So the canaliculi are in green. That's what is producing the bile. And then the portal triad is made up of these three major kind of vessels here. So the liver is kind of a really cool organ. It can regenerate itself. So if you lose a part of it, and to an extent, it can regrow on its own. More about the hepatocytes. They have lots of rough and smooth ER, Golgi apparatus, peroxisomes, and mitochondria. And that's because they're doing a lot of detoxification. Hepatocyte functions are to produce 900 milliliters of bile a day. They process bloodborne nutrients. Your liver hepatocytes will store glucose as glycogen and make different plasma proteins. They store fat-soluble vitamins, and they will perform detoxification. So they will convert ammonia to urea to be then excreted. Bile is a composition and enterohepatic circulation. It's a yellow-green alkaline solution containing bile salts, which will emulsify fats and absorb them. Bilirubin is a pigment formed from heme, from the breakdown of hemoglobin, and also cholesterol, triglycerides, and other things. Bile salts are what we call a part of the enterohepatic circulations. That means that this is like a recycling mechanism that conserves bile salts. So bile salts will be reabsorbed into the blood by the ileum. They're returned to the liver, and they're resecreted in newly formed bile. So that's called this enterohepatic circulation because it's only inside the hepatic or liver kind of circulation system. About 95% of secreted bile salts are recycled, so only 5% is newly synthesized each time. which is pretty efficient. So this just shows you how the bile salts are constantly kind of in this within this liver hepatic circulation. They'll be secreted into the duodenum and then reabsorbed back into the ileum in the hepatic portal vein, taken back to the liver to create more new bile salts. And again, bile salts are important for digestion of lipid and also absorption of lipid. Hepatitis is a viral infection, drug toxicity or wild mushroom poisoning and inflammation of the liver. Cirrhosis is a progressive chronic inflammation from chronic hepatitis or alcoholism. Your liver can become fatty or fibrous, kind of scarring with tissue. It could lead to portal hypertension. So because of the scarring or extra fat, it could kind of squeeze off or interfere with the circulation of the portal system, which are the veins that take the nutrients from the digestive tract. Liver transplants are successful, but livers are very scarce. Liver can regenerate to its full size in 6 to 12 months after about 80% removal. Alcoholism and alcoholics just really have a tough time with their liver. So that's the only soapbox I go on. If you know me, are smoking and alcohol, just please watch what you put into your body because you only get one liver, as well as everything else, but your liver is your friend. The gallbladder then, pretty much the gallbladder just stores and concentrates bile, and that's all I want you to know about that. Gallstones are caused by too much cholesterol or too few bile salts. They can obstruct the flow of bile from the gallbladder, and it can be extremely painful. Obstructive jaundice is a blockage. It can cause bile salts and pigments to build up in the blood, and that can result in yellowish skin. Jaundice can also be caused by liver failure. The gallstone treatment is to remove it, dissolve it. That's pretty much it. The pancreas is mostly retroperitoneal, so outside this peritoneum in the back. It's deep to the curvature of the stomach. It has a head and a tail. It has exocrine functions that produce pancreatic juice. The acini cells produce zymogen granules, and then ducts that secrete it to the duodenum. So this pancreatic juice is important for digestion. The endocrine process, or the endocrine function of the pancreas, is to secrete insulin and glucagon by the islet cells. Insulin lowers blood glucose levels. Glucagon raises blood glucose levels. So here are the acinar cells secreting enzymes. This is kind of one acinus as shown there. Here are the acinar cells and then the pancreatic duct. And again, the acinar cells are secreting the digestive enzymes. Here's a look at what makes up pancreatic juice and how much is produced every day. It's made up of proteases, digesting proteins, amylase for carbohydrates, lipase for lipids, and nucleases to break down nucleic acids. And this kind of takes you through those. I do want you to know kind of these main 4 classes of digestive enzymes that make up the pancreatic juice. Other than that, you don't need to know too much more of that. Let's see. This is where the pancreas, the pancreatic duct and the bile duct will unite in the wall of the duodenum and empty their contents through the major duodenal papilla. So bile from the liver and gallbladder travel through the common hepatic duct. This is actually a great picture describing the common hepatic duct going up, cystic duct going to the gallbladder, and then the common bile duct traveling down, uniting with the pancreatic duct and emptying all their contents, whether that's pancreatic juice and bile. Regulation of bile and pancreatic secretions, again, they're controlled by neural and hormonal controls. The hormonal controls are choleocytokinin, the CCK, and secretin. Regulation of bile and pancreatic secretions, pretty much they're increased when there's food or some sort of stretching of any sort of structure, or they sense that food is in the duodenum, then they get an increase to be secreted. Okay, and that's the end of this one. I think there's still one more, and we're going to try to get through that one too. If you guys are still with me, awesome. Thanks for your attention. Let me pull up part C here. Okay, so part C. And again, we're kind of just working our way from the top to the bottom in the digestive tract. Let me go ahead and share my screen so you're in me with that. Okay, So here we have part C. And we're working our way down to the small intestine. Your small intestine is the major organ of digestion and absorption. So not much has happened at this point. It's about 2 to 4 meters long, which is 7 to 13 feet, but it has a small diameter, which gives its name. The duodenum, the junum, the ileum. The duodenum is the smallest, but it does have the most features. And then the jejunum and the ileum joins the large intestine. So here's kind of a quick anatomy review of that. Again, you can pause um, the video if you need to review that. Here's a look at where the duodenum is connected to those major ducts that empty out their enzymatic secretions that we just talked about. Blood supply, different bloods carry oxygen-rich blood to the duodenum. Nerve supply, it's under parasympathetic control because again, parasympathetic is your rest and digest division. It's a little modified. It's microscopy of anatomy for absorption. Modifications include circular folds, villi, and microvilli. And your quiz includes some of this if you're still listening. The circular folds are permanent folds that force chyme to slowly spiral through the lumen, allowing more time for nutrient absorption. A villi is a finger-like projection with a core that contains a dense capillary bed and a lymphatic capillary called a lacteal for absorption. And microvilli is like a fuzzy, like brush border that contains membrane-bound enzymes used for final carbohydrate and protein digestion. So here's a look at kind of the small modifications, the structural modifications of the small intestine that help to increase its surface area for digestion and absorption. Here's the villi, the circular folds, the villi, and then the microvilli are like kind of just the brush border on the villi. And this picture is on your quiz. There's a matching question on your quiz that labels a picture that looks like this. We'll talk about what the lacteal does. This is a part of the lymphatic system, helps to absorb fat. You can see how the villi has a lot of blood capillaries, different goblet cells secreting mucus, other enterocyte cells. You have this, what we call intestinal crypts with Paneth cells at the bottom and different glands. You can see the microvilli makes up this little tiny brush border kind of on the epithelial side of each villus. And this is just a cool look at what each villus looks like. And then even another view at the microvilli forming the brush border. Okay, so enterocytes, here's the histology. You have crypts, which are tubular glands scattered between the villi. You have 5 types of cells found in the villi. Enterocytes, they function to absorb nutrients and electrolytes. The crypts produce intestinal juice, which is the watery mixture of mucus that acts as a carrier fluid for chyme. Goblet cells secrete mucus. Enteroendocrine cells are a source of the CCK and secretin, again, the hormones that help to control digestion. Paneth cells are found deep in the crypts, and these help to fortify the defenses. So they'll secrete some antimicrobial agents that can destroy bacteria. And then stem cells will just be continuously dividing to produce the other cell types. The histology of mucosa-associated lymphoid tissue that protects the intestine against microorganisms. You have Peyer's patches located in the lamina propria. I'm not going to ask you guys too much about this, but this is sometimes called MALT, mucosa-associated lymphoid tissue, and this just protects your intestine against microorganisms. And there's different kind of parts to that MALT that are important. Chemotherapy targets rapidly dividing cells such as cancer cells, and the negative part of this is that it also targets the rapidly dividing GI tract epithelium. Your GI tract has cells that are constantly dividing and replacing themselves because your GI tract is under so much abrasion that it constantly, every 2 to 3 days, needs to regenerate the cells and the lining of it. This is a reason why many patients who undergo chemo have a lot of symptoms of nausea, vomiting, and diarrhea because their GI tract is being just like destroyed by these chemotherapy. Just really tough on them. Intestinal juice. This is just the intestinal juice. So lots of it is secreted daily, almost 2 liters. It's slightly alkaline, and that's to kind of counteract the acidic environment that the food just came from in the stomach. Okay, sources of enzymes for the digestion. Digestive process takes about 3 to 6 hours to absorb all the nutrients and most of the water. We have bile, bicarbonate, other digestive enzymes imported from the liver and pancreas. Brush border enzymes from those microvilli are bound to the plasma membrane to perform the final digestion of chyme. Regulating chyme entry, chyme delivery has to be slow to prevent osmotic loss of water from the blood. The low pH of the chyme has to be adjusted upward. Remember, this chyme came from the stomach, so it's incredibly acidic. So the chyme is mixed with bile and pancreatic juice to continue digestion. That's why that major duodenal papilla, it empties out into the duodenum right when the chyme gets there for the stomach to try to make it less acidic. After a meal, segmentation is used, initiated by intrinsic pacemaker cells. It mixes and moves the contents. Parasympathetic, again, increases the motility of your small intestine, and sympathetic decreases. Peristalsis increases between meals, initiated by a rise in hormone motilin. Each wave starts distal to the previous wave, referred to as the MMC. Complete trip from duodenum to ileum takes about 2 hours. So again, a look at the differences between peristalsis and segmentation, and both are used in the small intestine. Ileocecal valve control, there you have a sphincter that will relax and admit chyme into the large intestine when you have a gastroileal reflex, and then it'll close when the chyme exerts backward pressure to prevent regurgitation back into the ileum. So this is just the control of the small intestine motility, things that will stimulate the effect and the mechanism. Here's an overview of the functions of the GI organs up to this point. And this is a great chart to review everything. There probably will be some questions from a chart that looks similar to this, but to be honest, if you study just these 2 slides in this whole chapter, you'll probably do pretty well from the questions on this test. Moving on to the large intestine, also known as the colon, it has 3 unique features not seen elsewhere. The Tenier coli are 3 brands of longitudinal smooth muscle that help to kind of segment and mix and move the feces along at this point. The haustra are pocket-like sacs caused by the tone of the Tenier coli. And then you have appendages, which are fat-filled pouches of the visceral peritoneum. Subdivisions of the large intestine, the cecum is the first part of the large intestine. The appendix is a mass of lymphoid tissue. It's part of that MALT system of the immune system. It's a bacterial storehouse capable of recolonizing gut when necessary. And the twisted shape of the appendix makes it susceptible to blockage. So otherwise, it really has no known function, but because it has all of this kind of extra bacteria associated with it, if it bursts, that can be very harmful to the surrounding tissue. The colon has several regions: the ascending, transverse, descending, and sigmoid colon, which you can review from anatomy. The rectum, you have 3 rectal valves that stop feces from being passed with gas, which is probably a good thing. And then the anal canal is just the last segment that opens to the body exterior at the anus. And it has 2 sphincters. Smooth muscle is under involuntary control, but the external anal sphincter is under skeletal muscle control, and that's something you can control. That's something I'm trying to teach my 3-year-old daughter how to control. She's still not potty trained to go number 2. We're working on it. So here is the anatomy of the large intestine that you guys can review on your own from that. And this is a look kind of at the final valves that will help excrete the waste. And again, the external anal sphincter is what is under voluntary control, what you can feel. Okay, the relationship of the large intestine to the peritoneum, most of it is retroperitoneal, except for the transverse and sigmoid parts. And you can kind of see that here in a cadaver-like model. Okay, and I won't ask you guys too much about this. This transverse mesocolon is what kind of connects the large intestine together. The greater omentum is the apron-like structure that goes on top of the large and small intestine. And this just takes you through the mesenteries of the abdominal digestive organs. Again, these are just different layers and membranes that cover everything. I won't ask you guys questions about this, but it's just interesting to see how everything is held together. Appendicitis is acute inflammation of the appendix. It usually results from a blockage by the feces that traps infectious bacteria. Most common in adolescence when the entrance to the appendix is at the widest. Um, Venous drainage can be impaired. Ruptured appendix can cause peritonitis, which is an inflammation of that peritoneum kind of membrane wall. Symptoms is a lot of pain, loss of appetite, nausea, vomiting. Treatment is surgical removal, or in some cases, extra antibiotics. The large intestine contains thicker mucosa. It does not contain any circular folds. And I think I'm going to skip over some of this. You can read about hemorrhoids. Superficial venal plexuses can form hemorrhoids if they get inflamed. Okay. And the bacterial flora, there's different types of bacteria in the large intestine. The metabolic functions of this are to help ferment indigestible carbohydrates and mucin, release irritating acids and gases, vitamin synthesis, keeping the bacteria in check. The immune system will destroy any bacteria that try to breach this mucosal barrier. So you kind of have to, your body has a way to keep that all in check as well. Gut bacteria and health. Mounting evidence supports findings that kinds and proportions of gut bacteria can influence the body weight, susceptibility to disease, and our moods. And manipulating your gut bacteria may become a routine healthcare strategy in the future. This is very interesting. You can look more into that, but that is kind of on the front lines as well as new research is being done on gut bacteria and gut health. Here are some imbalances of the large intestine: antibiotic-associated diarrhea, difficile. I'm going to skip over some of this. Digestive processes in the large intestine. So its main role is to reabsorb water. Residue will remain in the large intestine about 12 to 24 hours. No food breakdown occurs except what any sort of bacteria digest. Vitamins, water, and electrolytes are usually reclaimed and reabsorbed at this point. And the main function of your large intestine is just to move things along, move the fetus to the anus and then get rid of it. The large intestine is important for our comfort, but it's not essential for life. Many people who have colon cancer need to get parts of their ileum removed, and they can still survive. The motility of the large intestine is due to these contractions of the haustrum pouches. And these are mass movements. They're slow, powerful peristaltic waves that are activated 3 to 4 times per day. And then the descending colon and sigmoid colon will act as a storage reservoir. Diverticulitis is the presence of diverticula, which are kind of polyps or pockets. A low fiber diet can narrow and cause contractions that can increase the pressure on the colon walls that can cause these. Diverticulitis is when they get inflamed and they can rupture and leak into the peritoneal cavity, and that can be life-threatening. Irritable bowel syndrome is a functional GI disorder. It's painful, changes your stool, your feces, bloating, flatulence, passing gas, nausea, depression, and stress is usually a factor of that. Defecation is just the movement of all the feces out of the body through the anus. Conscious control will allow relaxation of that external anal sphincter. Muscles of the rectum contract to expel the feces using the Valsalva's maneuver, which helps to increase intra-abdominal pressure. Okay, and you don't need to know too much else about that. Diarrhea, watery stools, results when the large intestine does not have sufficient time to absorb the remaining water. Usually is caused by any irritation of the colon by bacteria. Prolonged diarrhea could result in dehydration. So vomiting, diarrhea, the big thing there is becoming dehydrated, but also losing a lot of your electrolytes, which could cause acidosis, loss of potassium. Constipation can occur if food remains for an extended period of time in your colon and too much water is absorbed. It may result from insufficient fiber or fluid in the diet, lack of exercise, or laxative abuse. The physiology of digestion and absorption. So this digestion breaks down ingested foods into their chemical building blocks, and only these molecules are small enough to be absorbed across the wall of the small intestine. So if you're still with me so far, this is what your final workshop will talk about. And I'm just going to go ahead here because I think I kind of want to get to The final workshop actually is the next chapter, excuse me. I got a little confused here. This is just going to take us through some of the digestion and absorption of different molecules. Pretty much digestion just breaks anything larger down into a monomer, one simple molecule. And enzymes will carry out what we call hydrolysis, where water is added to break a chemical bond. Absorption is just when the molecules are absorbed into the bloodstream or the lacteals. And this just shows you how the molecules have to go through the route of absorption is through what we call an enterocyte into the bloodstream. So this is just the absorption of molecules that have been broken down directly through the enterocyte, not through the tight junction, through the cell into the bloodstream. Processing of nutrients, only monosaccharides can be absorbed. So starch and disaccharides have to be broken down. That begins with salivary amylase, and they're eventually broken down into monosaccharides, which are single sugars like glucose, fructose, and galactose. And that's carbohydrate digestion. Steps of starch digestion in the intestine, pancreatic amylase helps to break down starch or glycogen. The brush border enzymes also help. And then monosaccharides, which again are single sugars, are transported across the cell membrane into the bloodstream. And this just shows how starches are broken down into monosaccharides, and they are transported through the enterocytes, which are the cells that line the GI tract, eventually into the bloodstream. That's all you kind of need to know, that general process. I won't ask you too many details about that. This is a little bit about lactose intolerance. They have deficient amounts of lactase and they cannot consume lactose. It remains undigested and creates a concentration gradient. Basically, that results in diarrhea because it's preventing water from being absorbed. So also not very fun to be lactose intolerant. The digestion of proteins, the source of protein not only is dietary, but also includes digestive enzymes and proteins from the breakdown of mucosal cells. Proteins are broken down into large polypeptides, and then finally, eventually into their amino acid monomers, which is again the single chemical kind of molecule. Digestion begins in the stomach when pepsinogen is converted to pepsin at a pH of about 1 to 2, and it becomes inactive in high pH of the duodenum. So pepsin is only active at low pHs in the stomach, and pepsin is important for protein digestion. Other pancreatic proteases that help to digest proteins are shown here. That's all you really need to know. I won't get to too many specifics about this, but proteins will be broken down via different enzymes into their amino acid components, which will eventually get secreted into the bloodstream. Digestion of fats has to do with emulsification. That's done with bile salts. And then pancreatic lipases will break fat down into their individual components. A chylomicron will eventually be enter into that lymphatic lacteal, and lipase also helps with the fatty acid degradation. So in terms of fat digestion, bile salts help to emulsify fats, and these bile salts kind of surround the fats. That's called emulsification. That just make them easier to be digested to form what they call these micelles. And the micelles just are more able to take that fat, cross the brush border, and be absorbed into the lacteal. So the important part here is we form these chylomicrons, and the chylomicrons are what will enter the lacteals and are carried away from the intestine and the lymph. So remember those the villi have the lacteals within them, and the lacteals are what basically are absorbing your fats that you eat and take it to the lymphatic system for absorption. Nucleic acids, so nuclei of ingested cells and food contain DNA and RNA, and you have different enzymes that break down nucleic acids into their component parts. Vitamin absorption, fat-soluble vitamins are carried by micelles and they diffuse directly into the absorptive cells. Water-soluble vitamins are absorbed by diffusion or by passive or active transporters, and vitamin B12 binds with the intrinsic factor and is absorbed via endocytosis. Absorption of electrocytes, again, this describes how different things get absorbed in the small intestine, but I won't ask you too much about this. Um, a lot of water, most of the water, 95% is absorbed in the small intestine just purely by osmosis, so it diffuses passively, and then most of the rest of it is absorbed in the large intestine. Malabsorption is anything that interferes with the delivery of bile or pancreatic juice, um, usually due to the damage of the intestinal mucosa by a bacterial infection or an antibiotic. Celiac disease is a common malabsorption disease. It's an immune reaction to gluten where it causes cell damage to the villi and brush border. So you can try to eliminate your gluten from your diet. So we got through this chapter. Thanks for listening, guys. I'll try to get the next chapter recorded. Thank you so much for your patience. I hope you guys are all staying healthy and well.